Okay, so everyone, welcome to session 2 of the 15th annual Showcase of Undergraduate Scholars. This is session 2 biology. I am Jessie Bowman, the student program and marketing specialist in the Office of Undergraduate Research. And before we get started with the second round of presentations, just a little housekeeping. This presentation is being recorded. Questions are encouraged from our audience as well as our student presenters. You all can actually verbalize your questions, but our audience will type their questions using the Q&A feature located in your toolbar. And this session we have this session has a moderator, Asia, from UK Libraries, and I'm going to let Asia introduce herself.
Hi there, I'm Asia Betancourt-McCarthy. I'm the STEM Instruction Librarian at UK Libraries, and I'm so pleased to be moderating this session. And we'll go ahead and get started. Kaylee, are you ready to go?
Yep. Go ahead and share my screen here. Hello, my name is Kaylee Meador, and my research for the past year and a half has been on the deletion of surf-like genes through CRISPR-Cas9 technology. And my research has been part of a bigger project that is the ongoing analysis of the Drosophila model of spinal muscular atrophy. And I work with Dr. Brian Ryman and Dr. Doug Harrison, who are two professors in the Department of Biology. So just a little introduction to understand why we are so interested in spinal muscular atrophy and how the genes that I research relate to this topic. Spinal muscular atrophy is the leading cause of infant mortality in the world, and there's currently no cure for this disorder. There's only treatments to try to ease some of the symptoms. It's a neuromuscular disorder, and it is caused by the lack of the survival motor neuron protein. So this protein ensures that the neurons stay healthy and live a long life, but whenever there is not this protein around, basically the neurons die prematurely, and this leads to neuromuscular symptoms such as not being able to exactly move your limbs when you want to, and eventually, like in the later stages of this disorder, people have difficulty breathing because your intercostal muscles are the ones that keep your lungs expanding and contracting to get air in and out of them, so it can lead to suffocation and death. So surf gene expression increases SMN protein levels. This has actually been proven by prior research done by Dr. Ryman's lab, and SURFs hypothesized to have a role in the homeostasis of the survival motor neuron protein. So with SURF helping with the longevity of the survival motor neuron protein, we decided to look at genes that were similar to SURF to see if they also affected the survival of this important protein. The genes that I looked at are CG15715 and CG18081. Those names are sort of a lot, so I'm just going to call them SURF-like genes from now on. They're genetically similar to SURF, and they have an unknown function. To the right here, you can see where the two genes are on the third chromosome of Drosophila, and there's also some more genes that are known just mapped around them to give you a sort of orientation of where you are on the chromosome. And lastly, we chose a Drosophila model of research because these genes are phylogenetically conserved. So the same genes that are present in humans are present in a degree in Drosophila. Our goals of my research project were to create mutations in the surf-like genes using CRISPR-Cas9 editing and genetic crosses, and then we screen these mutants through PCR and gel electrophoresis, and then we analyze the DNA sequences of the mutants in order to determine which ones we wanted to study further, and then we use the selected mutants in future studies. This slide just shows that the human surf-like genes are structurally similar to Drosophila, yeast, C. elegans, and other Drosophila surf genes. So the first row here on this is the human surf-like gene and the amino acid sequence of that. And then these two are the amino acid sequences of the surf-like genes in Drosophila that I researched. And as you can tell, these genes are remarkably similar, except for these two amino acids down here that differ. So due to this similarity, we believe that these two genes arose from a gene duplication event and that they may have a similar function. This is the overall genetic pathway of my crosses. So we started out with 3 separate Drosophila stalks that express certain genes in their germline to make the mutations inheritable. there was a CG15715 guide RNA, a CG18081 guide RNA, and then a CRISPR-Cas9, which was expressed on the nanos promoter. With CRISPR-Cas9, it's a genome editing tool that uses guide RNA to make double-strand breaks in DNA, and then through that and non-homologous end joining, when the DNA tries to repair itself, there are deletions or other mutations in the gene that may make the protein unstable and not viable. So since CRISPR-Cas9 was expressed under the nanos promoter in the Drosophila, it ensured that there were germline male mutations. So although the males themselves did not have mutations in them, their germline did, which made the CRISPR-Cas9 and guide RNAs inheritable for the next generation. And below each of these crosses are what we selected for in the offspring. So we use identifiable phenotypic markers of the offspring in order to know what genes these flies may carry. For example, curly wings, vermilion-eyed flies, and yellow bodies all gave us an indication of what genes they may have. And then to just ensure that it had deletions that we needed at the end, we screened the mutants through PCR and gel electrophoresis. We also use Down syndrome chromosomes, this TM3 right here on the second line in the bottom of this sort of fraction. It just ensures that there is no recombination during meiosis, and it allows us to follow the genes more clearly. And the end result was we crossed this final generation here to its siblings, and we made clonal mutant lines of the mutants of interest. This slide just shows how we screen the mutants through gel electrophoresis. We screened about 200 mutants by PCR and then running gels on them. So this first well right here is just a marker to show where some of the nucleotide bands would be. And then this W1118 is wild type for the CG18081 gene. And then here you can see that all these wells have the fly marked by the gene and the number of the fly that is assigned to them. And then some have HZ and some don't. The HZ stands for homozygous. So that means that this fly is a deletion mutant in both copies of the allele. A good example of this is the CG1808120 here. The heterozygous one, you can see that this middle band is the wild type allele for 18081. This bottom band is a deletion mutant. And then this band right here is actually an interesting case of heteroduplex DNA that we believe formed during the gel. And it may have formed like a bubble where the DNA was binding to itself, and it caused it to migrate more slowly through the gel. And then in the homozygous, you can see that there's only the deletion mutant because there's a double copy of that allele. There's about a 25% success rate in the creation of these mutants, and with one exception, all isolated deletion mutants were viable as homozygotes. And the fact that almost all the mutants were viable suggested to us that maybe CG15715 was compensating when CG18081 was deleted, and vice versa. So for future experiments, we're thinking about deleting both of these genes to see what the phenotypic variation is. So after we confirmed that our deletion mutants had the deletion that we may want, we decided to have them sequenced off-site through ACGT Inc., and they sent us back a nucleotide sequence. And then we inserted that into programs on the internet that used all 6 possible reading frames, and we decided which one was most likely the reading frame used by the fly's cells. Then we converted that DNA sequence to an amino acid sequence and compared it to the wild type amino acid sequence available in flybase.org. And to the right here is just sort of our overall notes on the flies and where the mutations occurred in each fly. And in the end, we concluded that flies CG1552 and CG1820 were selected for additional analysis. So this just means that the 52nd fly that we tested for the CG15715 and the 20th fly we tested for the CG18081 were the ones that we believed had either the largest mutation, the mutation that had a significant frame shift, or the mutation that had both of those in the exon of the gene, so it would affect the overall expression of the protein. So in conclusion, genes CG15715 and CG18081 are 2 surf-like genes that are phylogenetically conserved between Drosophila, human, and other species. They have an unknown function, and there's a possible influence in proteostasis, especially in the cerebromotor neuron protein, which relates to spinal muscular atrophy. The CRISPR-Cas9 mutagenesis and genetic crosses were successful at about a 25% success rate, which is a lot higher than what we expected originally. And the fact that there were viable homozygotes throughout the experiment showed that these 2 surf-like genes may be compensating for each other in some way due to the fact that their amino acid sequences are so similar. And for future studies, we will use these mutants to investigate gene function. So as I mentioned before, we're interested in deleting possibly both surf-like genes and seeing how that affects the longevity and ability of the fly, or also deleting surf itself and the surf-like genes, just different combinations to see what the functional purpose is of these genes. And I will take any questions at this time.
Thank you, Kaylee. That was fantastic. If you've just joined us, please go ahead and enter any questions in the Q&A function at the bottom of your screen. And while we wait for those to roll in, I have a question. I was curious, what was the most exciting part of conducting this research for you?
I think the most exciting part for me was just working with my mentor a lot, because I actually got like a one-on-one experience, especially through Bio 395. I was the only student that he had in his lab. So I was able to work with him. Like every time that I went in the lab, he was there and we would talk about what our next step was on this. And then also he's a genetics professor in the biology department as well. And I was in honors genetics at the time. So he was able to help me understand my genetics better. It was just an overall good experience to be that close to a professor.
That's fantastic. And are you thinking of continuing this research moving forward?
Well, actually, I'm graduating this semester and I'm going to pharmacy school in the fall. So I might not be able to, but I definitely would if I could.
Fantastic. I'm going to give it just one more minute. And with that, thank you so much.
Thank you.
Our next presenter is Gavin. Gavin, are you all set?
Ready to go. Oh, I'm muted. Sorry. Can everyone see my screen?
Yes, we can.
Awesome. All right. So, hi, my name is Gavin Norman. I'm a member of the Lennon Lab, and I'm doing my presentation today on assortative mating in 2 sympatric species of pine sawflies. So the question is, why study assortative mating? Assortative mating is interesting to study because it is an important barrier that keeps diverging lineages apart and completes the speciation process. For this experiment, we're looking at how assortative mating is keeping the species Neodyprion pinetum and Neodyprion lecontii from mating and producing offspring with each other when they're in sympatry or the same geological location as each other. Sorry. And so to study this, we recorded videos of multiple trials that consisted of 2 arenas, as you can see on the left-hand side. Each arena was divided into 6 equal sections. In each section, a small petri dish with 1 male and 1 female was placed. In each arena, the females were all the same species, and there were 3 conspecific and 3 heterospecific males. And each trial lasted approximately 2 hours. So via videos and actually rearing these species, we collected a variety of pre-zygotic and post-zygotic data. We conducted 3 experiments, but today we will only be talking about the experiment that I was personally in charge of, and that was experiment RID, which was with Kentucky lecontii and pinetum from Michigan. So it's also important to point out we preserved all the adults using our assays for genetic analysis on a later date, if need be. So our first question we asked was whether there is assortative mating between lecontii and pinetum in each population. And so we conducted a Fisher's exact test, and this is our p-value. We're considering any p-value less than 0.05 to be significant, and therefore looking at these values, we can conclude that there is significant RI between the 2 different populations in experiment RID. And so all of this really means that we found there's something keeping the 2 species from mating with each other more frequently than they do themselves. But why is this, and which sex is responsible for these results? So we then looked at whether there are differences in the number of male attempts by cross type. Ultimately, we wanted to know if males make less attempts with heterospecific females. On the x-axis are the cross types: pine lecontii, pine pine, pinetum pinetum, lecontii pinetum, lecontii lecontii, and ultimately, and on the y-axis, the number of male attempts. And we conducted a Kruskal-Wallace test, and this is our p-value for this experiment. This p-value shows that the males are not picky and that the mating outcomes might be determined by the females. So we thought that they might be using size, since that is a common metric that insects use when determining if they will mate or not. And so to address this question, we took 5 different measurements from all males and females used in our no choice RI assays. Please pop up. So, sorry. Specifically, we measured body length, head width, thorax width, forewing length, and forewing width. Then for each of these 5 categories, we took the male measurement and subtracted it from the female measurement. And this gave us a difference measurement for each trait. So we broke our overarching research question down into several specific research questions, which we will go over individually now. Please pull up the graph. There we go. And so specifically, we first need to determine which sex determines the mating outcome. So far, we've begun to dissect male behaviors to get at this question. Now we need to determine what traits are being used to make mating decisions. And so we focused on size assortative mating first. When we were then curious to see if there was evidence of size assortative mating by body length when all the cross types were grouped together. Again, on the x-axis is mating outcome, where 0 is did not mate and 1 is mated. And the y-axis is the difference in size between the females and the males. We conducted a two-tailed t-test, and this is our p-value. Looking at the p-value, we can conclude that there is size assortative mating by body length when all cross types are considered together. And so this means that perhaps when we looked at each cross type individually, the p-values were not significant because of smaller sample sizes. But in most cases, p-values were approaching significance. Therefore, maybe these analyses will yield significant results after we do another round of RI assays in this upcoming semester and increase sample sizes. So we also had some follow-up questions from this. So one thought was that we had is that variation in the body size difference with different populations could be driving differences in mating outcomes. Therefore, we were curious to see if males differed in body length and if this varied across space. And so on the x-axis are the 2 species, the y-axis, the body length of the males. And we conducted a two-tailed t-test, and this is our p-value. Looking at the p-value, we can conclude that there is no difference in body length between Kentucky lecontii or Michigan pineatum. And so now what about females? So we wanted to see if females differed in body length and if this varied across space. And so on the x-axis are the 2 species, the y-axis is the body length of the females. And we conducted a two-tailed t-test again, and this is our p-value. Looking at the p-value, we can conclude that there is no difference in body length between lecontii from Kentucky that are female or Michigan that are pineatum females. This honestly surprised us because we thought that the pineatum females would be significantly smaller than the lecontii females. And so this suggests that females mate assortatively by size in general, whereby like lower quality, smaller females are more willing to mate with any male regardless of their species, but they can't use size alone to discriminate against heterospecific species. And so maybe they're using genital mismatch. And this is a follow-up experiment we'd like to do in the upcoming semester, and probably chemosensation, but we most likely are not going to test that. And so obviously we need to do more tests. In this upcoming semester, there will be projects in our lab looking at genital mismatch between males and females of different species to see if this is what is driving assortative mating in the RI barrier. And that is everything I have. Stop sharing so I can see this.
Fantastic. Thank you so much. Again, if you're a participant and would like to ask a question, please enter it in the Q&A function in Zoom. And as we wait for those to come in, it sounds like your research yielded a lot more questions sort of from your initial question. And I'm curious, during that experience, if you had sort of a takeaway that you could point to from the research experience and going through this process, what might that be?
So I've been in the same lab for about 3 years now. And if there's one thing that I can say about research, it's after you do an experiment, you might learn 1 or 2 things, but in the end, you realize that there are like 30 different variables that you need to test in the future. So like, it's kind of like, I don't know, it's hard. It's weird. Like you learn something, but then you learn that you don't know everything and that you need to keep doing research to actually get anywhere.
That is such a valuable insight. It's been my experience as well.
Definitely.
In addition to that, could you speak briefly into sort of what got you involved in the research? You said you've been in your lab for a number of years.
Yeah, so I was in STEMCAS freshman year, and I did a Bio 199 project with Dr. Weisrock. But the semester before that, I took a class with Dr. Weisrock. It was Bio 198 honors, and I don't know, it just really opened my eyes to like what working in a lab is like, and like what a really close relationship with the professor is like. And I was like, dang, I really want to do more of this. And so I went looking around for professors and stuff, and in Dr. Weisrock's lab is a joint lab with Dr. Lennon's lab. And so that's kind of how I came in contact with Dr. Lennon. And so I ended up joining her lab. It's been a really fun trip.
Fantastic. And we do have a question in the Q&A, which is, how has your research evolved over time?
Okay, so I'm sure this varies for different labs, but really what happened was my first year in the lab, I spent a lot of time just kind of like going through what was already known, what had been done, learning the system, how sawflies worked. Their genetics are weird. It's a haploid diploid system where males only have one chromosome and females have two. So it took me a while to just figure out what is already known. And then from there, I started asking more and more advanced questions, I guess you could say. So like it started off with something basic, like what happens when you feed different hosts to sawflies, and then it was not really that basic, and then it's moved on to this reproductive barriers.
Fantastic. Thank you again.
Thank you.
Okay, and our next presenter is Frannie. Are you all set? Francis?
Hi, yeah, I'm all good to go. Okay, so hi everyone, my name is Frannie Salisbury. I am a senior biology major, and I'm going to be presenting the research that I've been working on for the past 3 years, and then also a little bit about the capstone project that I'm doing right now. So the title of my project is An Exploration of Sleep Fragmentation and Sleep Enhancement on Alzheimer's Disease. So this is my poster, but I'm going to break it down on each slide. So I'm going to start out by talking about the sleep fragmentation study. And this is something that my lab has been working on for the past 3 years, and it's still ongoing today. So a little introduction. So the research I work on focuses on Alzheimer's disease. And Alzheimer's disease is a neurodegenerative disease that can be defined by amyloid beta plaques, neurofibular tangles, and neuronal death, all of which cause neuroinflammation. It's basically a disease that causes profound memory and cognition loss, as well as personality changes. It's a disease of the brain, and it mainly affects the elderly. Wait, let me go back. So it's believed that that Alzheimer's disease is affected by these increased amyloid beta plaques that are found in the brain of Alzheimer's patients. Amyloid beta is a protein, and it's normally found in the brain, but it gets cleared out as waste. But with Alzheimer's patients, it's seen to build up. It doesn't get cleared out as well. It forms these plaques, and then that ends up causing neuronal death, which then causes the cognition errors that are seen with patients. And so one theory is that the decreased clearance of amyloid beta is due to decreased sleep, because Alzheimer's patients have also been seen to have frequently disrupted sleep. They'll wake up in the middle of the night, they'll take naps during the day. So they just don't have an overall normal sleep cycle. And so basically this study wanted to investigate whether this sleep fragmentation is affecting the progression of the Alzheimer's disease and the buildup of amyloid beta in the brain. So this study basically investigated whether chronic disruption of sleep-wake cycles via sleep fragmentation increased amyloid beta 40 and 42 levels, which are just two different sizes of the amyloid beta protein. In the hippocampus of transgenic AD mice, we used mice that were 3x TG-AD, which just means they had 3 genetic mutations that caused the mice to have Alzheimer's disease. And so basically the hypothesis was that sleep fragmentation will increase the amyloid beta levels and neuroinflammation in the brains of transgenic mice, thus accelerating the progression of Alzheimer's disease. So how we went about this was we used all females, which the project has progressed from, like, this semester they're using both females and males, but the results I'm going to be presenting are just female mice, 8-month-old, and they were exposed to a 12-12 light dark cycle. Basically, we sorted these mice into 2 groups. One group was called undisturbed sleep, and they just had, lived their life like normal. And then the other group was a sleep fragmentation group. And the sleep fragmentation group underwent 4 sleep fragmentation sessions throughout the day. They were 1 hour long, and we did that 5 days a week for 4 weeks. So the sleep fragmented mice were basically kept awake for an hour 4 times throughout the day, because the daytime is when mice normally sleep, while the undisturbed sleep group did not have any disruption to their sleep. And then after the 4 weeks, all the mice, their brains were extracted, and then we basically quantified the amyloid beta amounts in all their brains to look at whether there was more amyloid beta in the ones that were sleep fragmented compared to the undisturbed group. So here's some results. The top graph is basically just showing the sleep data that we collected. And so those gray bars show the sleep fragmentation sessions throughout the day, and the blue line is the sleep fragmented group. And so what this graph is showing is just basically that we successfully sleep fragmented the mice, and that they were getting less % sleep during those sleep fragmentation hours when compared to the undisturbed group. Then these bottom graphs show the results from the quantification of the amyloid beta. And basically what we found, we used like 2 different solvents in order to quantify the data, and we found significant results that the blue bars, which is the sleep fragmented group, had significantly higher levels of amyloid beta 40 and 42 in the hippocampus when compared to the undisturbed group. And so that's basically just showing that there was a significant amount more of amyloid beta built up in the brains of the mice that were sleep fragmented. So the conclusions we got from this study was that the 3x TG-AD mice were a good model for Alzheimer's disease patients. Additionally, the sleep fragmented mice had reduced sleep during sleep fragmentation intervals, so the sleep fragmentation worked. Protein levels of A beta 40 and 42, as well as neuroinflammation markers, were significantly higher in sleep fragmented mice, leading to the progression of Alzheimer's disease pathology. And decreased sleep/shortened fragmented sleep cycles may play a role in the amount of amyloid beta 40 and 42 cleared from the brain, resulting in increased deposition in the hippocampus. And then if future rodent studies support these findings that chronic sleep fragmentation advances Alzheimer's pathology, then improving sleep consolidation would be potential therapeutic strategy for reducing the progression of Alzheimer's disease patients or neuropathology in humans. And so that was basically the left side of my poster. The right side is what I'm currently working on right now, which is a branch of this study. So basically, from the last study, we found that sleep fragmentation made Alzheimer's disease worse. So we wanted to look at whether a way to enhance sleep in order to reverse this. So the introduction, basically, if sleep fragmentation progresses AD pathology, then sleep enhancement may be a way to slow AD pathology. There's pharmaceutical options for increasing sleep. However, these come with side effects and often have not been shown to actually improve sleep disorders, only treat symptoms currently. So we wanted to look at a mechanical / more natural way of enhancing sleep. So how we did this was using a it was called the mouse Quake system, but it was basically a platform that rocked the mouse cage, kind of like a mom would like rock a baby to sleep. That was the like basis of this idea. And so the hypothesis was that slight rocking will improve sleep time, latency, and sleep out length in mice. So basically what I did was I used a 4-day protocol. Day 1, I put the mouse in this cage, the mouse Quake cage, along with a piezo sleep monitor that allows us to monitor how much they're sleeping. Day 1 was just acclimation, so I didn't analyze any data. Day 2 was baseline. Day 3 was experimental, where the platform rocked at 2 hertz frequency for 12 hours, so 7 a.m. to 7 p.m., which is lights on to lights off, which would be the time that a mouse would be sleeping. That's like nighttime to them. And then day 4 was recovery day, where there was no rocking, and I just wanted to record the sleep and see what happened after they were rocked. What I found, so this data is only from one mouse because I am basically in the middle of this project right now. I have my 3rd mouse in the shaker like right now. But the first mouse, we did not find any significant results. So we didn't find that using the mouse Quake system improved sleep at all. I looked at total sleep as well as light phase sleep, and there wasn't anything significant there. So honestly, some conclusions from this is that due to the small sample size, we can't really make any conclusions. As this is an ongoing study, the continuation of this project with more subjects is crucial, which I'm currently working on. Additionally, the failure to find significant results may be to the fact that the mouse did not have a sleep disorder before. So unlike the Alzheimer's disease mice that already had like issues with their sleep, that they might have seen a bigger like change with enhancement, whereas someone who already sleeps normally, enhancing it probably won't do as much because it already sleeps normally. Additionally, the equipment I'm using is like relatively prototype, so it has some like blips and issues which may be affecting the results. So additional trials are being conducted currently, and the future goal of this study is to determine that rocking can enhance sleep and test this on the Alzheimer's mice to see if this could slow the progression of Alzheimer's. And here is my poster one last time, and that is all I have if there's any questions.
Thank you so much. As a reminder for our audience, please enter your questions in the Q&A. And to get us started, one just came in, so I'll ask that one first. It says, human moms rock their babies, but do mice do anything like this with their young that would lead you to consider this work?
So I don't think this is like something that has been seen in mice. Honestly, the ultimate goal would be for this to be used on humans, just using mice as a subject. The basis of thinking that slight rocking might enhance sleep was basically with the moms rocking babies, and then also if you think about people like in a car or on a boat, like motion tends to like induce sleep more. So it wasn't necessarily like, oh, we think that sleep or this would enhance mouse sleep, but it's more of just to see if in general this would enhance sleep in the future to be used on human patients.
Fantastic. And another question from the Q&A is, how has your research affected your own sleep habits?
This is honestly a very good question because it has affected my sleep habits a lot. I was definitely someone who was a late nighter, pulled those all-nighters before an exam. But after working in this lab for so long and seeing how 4 weeks of disrupted sleep on a mouse can have such an effect on their brains, it definitely scared me a little bit. And so I'm getting my 8 hours every night. That's something that I value a lot more now. And I think everyone should value sleep a little bit more because I definitely didn't. I know college things seem super important and sleep seems like it goes on the back burner, but it's definitely important. And I have changed my mind a lot from researching this.
Fantastic. Thank you again. Our next presenter is Claire. Claire, are you all set to go?
Yes. Share my screen. All right, so do you see my poster currently?
I do.
Perfect. So hi, everyone. My name is Claire Scott, and I am a senior in the Jeremiah Smith lab here in the Department of Biology. So my project focuses on the effects of histone methyltransferases on programmed genome rearrangement in sea lamprey. So sea lamprey, they look kind of like an eel. They are jawless vertebrates that diverged from the vertebrate lineage about 550 million years ago, which can sound like a lot, but it allows us to use the sea lamprey as an evolutionary model to study genomic mechanisms throughout the vertebrate lineage. So comparing that to today's vertebrates. Lamprey do something really interesting with their genome in that they undergo programmed genome rearrangement, which allows for the deletion of about 21% of their genome in somatic body cells, but then that genome becomes germline specific. So it's only kept in the germline cells. This happens throughout the cell cycle. So you can see here in the top left, it shows that the red DNA, which is eliminated, in anaphase becomes lagging and then becomes packaged into micronuclei, while the retained DNA, which is shown in blue, becomes packaged into the nucleus. These micronuclei are then degraded by about day 3.5, and this begins about the 7th cell division. So by the end of day 3, this process is essentially complete. So the goal in our lab, one of the goals in our lab is to figure out how programmed genome rearrangement takes place. So my piece in this puzzle is relating it to epigenetic mechanisms. So lamprey undergo epigenetics, as well as today's mammals. We ourselves, our body uses the silencing mechanism. Particularly, I'm looking at methylation of histones. So histone 4 lysine 9, or histone 4 lysine 20 becomes methylated, and histone 3 lysine 9 also becomes methylated, and that allows for the silencing of that genomic portion. So those genes specifically are SUV420 that codes for the methyltransferase for histone 4 lysine 20 methylation, and then the SUV39H1 gene, which codes for the methyltransferase for H3K20 methylation. So by looking at these genes, we will look at knockout embryos through the CRISPR-Cas9 system, as well as their control siblings, to compare the amount of micronuclei in light sheet imaging. So to begin this process, we started by having to clear these embryos out so that we could actually obtain the light sheet images. So in the bottom left, you can see before we do this procedure, the embryos are very opaque and dense because their yolk is just, you can't really see through it. So then after this optimized protocol, on the right, you can barely see the embryos. So that is ideal so that we can see the nuclei in the embryo. So once we apply the stain and then embed these embryos in gel, and then you just send it through a light sheet microscopy machine. And so then it just sends a sheet of light through the embryo, and then we get this nice 3D image here, which is in the middle top. So we have a 3D image of the embryo, and you can even see how the nuclei are distributed throughout. So they're more dense towards the middle and then radiating outwards. One of the grad students in our lab has developed a pipeline that allows us to analyze the number of nuclei and the number of micronuclei per each nucleus. So then that gives us a measure of the programmed genome rearrangement that is taking place in each of these embryos. And these embryos are sampled at day 2, so right in the middle at the peak of programmed genome rearrangement. Now in these graphs in the bottom, you can see that the SUV420, so this is the top graph, we see an increase in the amount of micronuclei in knockout embryos. So when you don't have this gene, so you don't have this methyltransferase, putting the silencing mark and regulating that, you get an increase in this level of programmed genome rearrangement compared to controls. However, in SUV39H1 knockouts, you see about the same level of micronuclei per nucleus throughout these day 2 embryos, which is very interesting because they both have a same epigenetic silencing mechanism within the cell. To further validate these results, I performed immunohistochemistry, which is in the top right. You can see that for a control embryo, you have a micronucleus shown by the red arrow here. And then to look at the antibody itself and see if that has been stained and that you have that trimethylation mark that's in the bottom here under this GFP filter. We saw that by knocking out this gene, SUV39 and SUV420, you get a significant decrease, a statistically significant decrease in the proportion of micronuclei that have these positive signals for trimethylation. So that shows us that by knocking out this gene, you have a significant decrease in the signal itself, the trimethylation of these spots. And also another interesting thing that we found with this was that, so SUV420, there's 3 different types of the gene. We suspect that one is involved in monomethylation, one is involved in dimethylation, and one is involved in trimethylation due to the proportions of positive signals from each being approximately equal, which is not what we expected. So that's what I'm currently working on, is trying to use 3 different fluorophores for each antibody on one slide for each knockout to see which gene is involved in which methylation mark. So yeah, so after all of this, I'm working on also doing real-time PCR to figure out a quicker way to screen all of these different genes. As you can see that this, to screen for these 2 genes, is quite a lot of work. So we're working on that as well, and just further looking into the distribution of all the micronuclei throughout the embryo to see if there's a difference in the distribution and not just the counts. And I'll take any questions now.
Fantastic, thank you so much. If you're just joining us, please go ahead and put any of your questions in the Q&A box. And while we're waiting for those to roll in, I had a question about what motivated you to pursue this particular research project?
Yeah, so it's kind of obscure, kind of out there. I mean, you don't think of lamprey when you think of research, you think of mice. But I came into college really interested about epigenetic mechanisms and just how the genome is able to be so different in different people, but you have the same basis of genes. So then I found this research. I just looked through the biology website and I was like, this is really interesting, talking about different types of genomes. And then I was able to, I talked with Dr. Smith and I was able to bring in the epigenetic portion and work on kind of both at once. So it's really interesting.
Fantastic. And we got another question that reads, do you have any idea whether the increased level of DNA rearrangements seen in response to methyltransferase mutation reflects an increased efficiency for deletions at normal sites or spreading the deletions to different chromosomal segments?
So I haven't really looked at exactly the deletions in the different chromosomal segments. We're thinking that it's an increase in the efficiency of the deletions at the normal sites. So it's affecting the significance and how you're able to do programmed genome rearrangement. And then we've then done RNA-seq afterwards to see where these deletions are occurring, and they're occurring at those same sites. So it's going to be an increased in efficiency of that rearrangement.
Fantastic. Thank you so much. This was wonderful. Our next presenter is Madison. Madison, are you all set?
Yeah, I am. Can everyone see that?
Yes.
Okay. Sorry, yes, you can.
Good. All right, so hello everyone. My name is Madison Webb, and today I'm going to be talking to you about some of the work that I've been doing in Dr. Osborne's lab regarding the impact of CBD on pulmonary function in African green monkeys. So first, just to give a little bit of background, the endocannabinoid system is a biological system that's highly conserved among vertebrate species. It's composed of endocannabinoids, which are endogenous lipid-based neurotransmitters that bind to cannabinoid or CB receptors. And these CB receptors are expressed in both the central and peripheral nervous systems. And cannabidiol, or CBD, is an exogenous antagonist of CB receptors. And also importantly, for my research, it's a known allosteric modulator of CB receptor action. And CB receptors themselves are involved in inflammatory responses in peripheral organs, including in the lung tissues. And CBD administration has been shown to reduce airway inflammation and fibrosis in vitro. However, little is understood regarding the relationship between pulmonary disease and CBD modulation of general CB receptors in vivo. And the African green monkey, or AGM, has bronchiolar tissue and tracheal branching similar to that in human lungs, which makes it a really valuable translational model for the study of CBD treatment on pulmonary fibrosis-related lung dysfunction. So as a result, we hypothesized that administration of NCMB1, which is a CBD-containing drug derived from hemp, will increase measures of pulmonary function in AGM with previously induced pulmonary fibrosis. And these measures of pulmonary function that we use include respiration rate, inspiratory flow rate, and tidal volume. So in order to test this hypothesis, we took 16 male AGM and took baseline tidal volume, inspiratory flow rate, and respiration rate measurements. And we divided these 16 AGM equally into 4 groups. First is vehicle, second was an undiluted or 1 times dilution of NCMB1, third was 5 times dilution, and fourth was 25 times dilution. And drug was administered, or lack of drug also was administered via banana sections twice daily for 2 weeks. And then after 1 week of treatment, we took tidal volume, inspiratory flow rate, and respiration rate measurements. And then we took these measurements again after a 2nd week of treatment. So in doing this, we found that tidal volume is increased in AGM that were given the 5 times dilution of NCMB1 after 2 weeks of treatment. And we also found that inspiratory flow rate was increased in AGM given both the 1 times and 5 times dilution of NCMB1 after 2 weeks of treatment. And then lastly, we found that respiration rate was increased in AGM given both the 1 times and 5 times dilution of NCMB1 after both 1 week and 2 weeks of treatment. So in conclusion, all 3 measures of lung function increased relative to baseline in AGM given a 5 times dilution of NCMB1 for 2 weeks. And then 2 measures, inspiratory flow rate and respiration rate, increased from baseline in AGM given undiluted NCMB1 for 2 weeks. So these results suggest that CBD might be a viable treatment to improve lung function in individuals with lung disease, such as COPD. And 5 times diluted NCMB1 in particular appears to have the most therapeutic effect. And currently we're in the process of performing immunohistochemistry on the hypothalami of the AGM used in this study using CB receptor antibodies, because we feel like it's possible that sensory afferent signaling could lead to changes in the distribution or the concentration of CB receptors post-treatment with NCMB1. And additionally, some other possible future studies that we're interested in is treating the AGM with 3 times and 8 times dilution of NCMB1 so that we can better pinpoint the ideal therapeutic concentration of drug for the greatest improvement in lung function. And we're also interested in doing a chemical analysis of the NCMB1 compound using some sort of technique like gas chromatography, mass spectrometry, so that we know exactly what's in the compound and in what concentrations, so that we might be able to better figure out what's having the therapeutic effect that we're seeing. So with that, yeah, I'm done, and I'm more than happy to take any questions that you guys have.
Fantastic. Thank you. While we wait for questions to come in, I am curious how you kind of develop this research question and protocol that you uncovered?
Yeah, so Dr. Osborne works with African green monkeys, so it's a model that he uses almost exclusively. So, and this is actually contract work that somebody had requested of him. And because of my interest in chemistry, I'm a chemistry major and also a neuroscience major. So because of my interest in those two things, we kind of thought it would be a good fit for me to be working on this project because I'm interested in sort of mechanisms of drug action and the changes that this may have on the brain. So that's why I got involved in this project.
Fantastic. And it sounds like your initial results or your initial study produced some kind of exciting results. Can you speak to maybe the most kind of exciting or interesting part of this study?
Yeah, well, it's interesting because you see CBD, floating about, and especially I feel like recently it's been touted as some sort of like miracle thing for treating all sorts of diseases. I think some people like even say like it can cure cancer or whatever. But and although we haven't done a lot of research on it, it is exciting to have done this and see that it does have an impact at least on pulmonary function, which is really interesting and could possibly be a really good treatment for people with restrictive pulmonary diseases like COPD or even maybe like asthma. So it is interesting to see this thing that's been like, out in the public. You can see it at the mall and everything. And to see how that actually works in a living system is pretty cool.
Fantastic. And just one more minute. Does anyone have, if you have additional questions for Madison, put them in the Q&A and we'll get to them. I have one final question, which is if you could speak briefly maybe to kind of how you got into or became interested in doing this kind of research in the first place here at UK.
Yeah, well, similar to, I think Gavin was talking about how he was involved in STEMCAT. So that was my first research experience. And I was actually working in a neuropsychology lab with Dr. Mark Prendergast. And we were doing stuff with drug treatment. We were looking at alcohol exposure in rat models. So that got me interested in sort of the mechanisms of drugs. And so I reached out to Dr. Osborne and got involved in his work. And I know that he was doing a lot of stuff, especially lately with CBD and the treatment of, especially he's looking at all sorts of things, but lung function and stuff like that. So I thought it would be a really good fit and it's been a really great opportunity.
Fantastic. And we have one more question, which is great presentation. What are your future career goals and how has research impacted your college experience?
Yeah, well, I came into college pretty much knowing that I wanted to have a career in scientific research. So, sorry, that email thing distracted me. So doing research has pretty much in college has pretty much cemented that's my career goal. So my future plans after college are to get my PhD. I'm hoping in neuroscience, but I'm also looking at some biochemistry programs as well. I'm not sure exactly what's going to be the best fit for me, but I'm currently going to take a gap year and do some more research because I like it so much. So yeah, it pretty much has just cemented and given me more confidence in the fact that that's something that I want to do with my future.
Fantastic. Thank you so much. And then our, I believe, final presenter is Cameron. Cameron, are you all set to go?
Yep. Just share my slides here. Okay, so hi everybody, my name is Cameron Rostron, and I do research in Dr. Julie Pendergast's lab, who is one of my amazing mentor, and the other one is Bukola Amatwala, who is an amazing graduate student in our lab. And today I'm going to be talking about my research in the regulation of circadian rhythms by G protein coupled estrogen receptor 1 in female mice fed high-fat diet, which I know is a really specific title, so I'll talk a little bit more about each of those before I get into the results. So first, I want to talk a little bit about the circadian system. So circadian rhythms are 24-hour cycles that regulate physiology and behavior in all mammals and many other organisms, but specifically in humans and also in mice, which is the model that I used to look at this research. And circadian rhythms entrain to the light-dark cycle of the environment via the retina of the eye, which takes in light from the environment and then passes this information back to the suprachiasmatic nucleus of the hypothalamus. And this brain region is the main pacemaker of the body, and it also coordinates the timing of the circadian clocks in the peripheral tissues throughout the body. So previous studies, both in our lab and in other labs, have shown that male mice fed high-fat diet experience disrupted circadian rhythms as a result of high-fat diet feeding, and these disrupted rhythms contribute to the development of diet-induced obesity. However, female mice fed high-fat diet do not experience circadian disruption. So they have normal circadian rhythms, and this contributes to their protection against diet-induced obesity. So even on high-fat diet, female mice remain lean. And previous work in our lab that was recently published found that female mice are protected because of the presence of circulating estrogen. So estrogen is the factor that's protecting these circadian rhythms from high-fat diet feeding disruption. So I hypothesized that G protein coupled estrogen receptor 1, or GPER1, is necessary to protect the daily rhythms in female mice from disruption by high-fat diet. So GPER1 is a cell surface receptor, which when estrogen binds, it causes a cascade of secondary messengers, which cause a variety of effects within the cell, and those typically take place within a few minutes of binding. So it's a very rapid acting receptor. So to test out this hypothesis, I used wild type and GPER1 knockout female mice, and they were fed chow from birth until 7 weeks old, at which point we single housed them and switched them to a low-fat diet with 10% of the metabolizable energy from fat. So they stayed on this diet for 1 week, and then we switched them to a high-fat diet with 45% of the energy from fat. And we chose this diet because this corresponds to the fat content of the average American diet, which is pretty high. And then we continued the high-fat diet feeding until 14 weeks of age, when we measured body composition, fasting blood glucose, and cultured the tissues and measured molecular rhythms in these tissues for some mice. And then throughout the 7-week experiment period, we measured eating behavior and locomotor activity rhythms at a few different time points to look at how those differ between the groups. So the first thing that we found is that the female GPER1 knockout mice, which are the red icons here, gained more weight when fed high-fat diet than the wild type mice over the course of the experiment. And one possible reason for this might actually not be circadian at all. We also found that they consumed more food over the course of the experiment. So you can see here, starting on high-fat diet, the GPER1 knockout females consumed more food than the wild types. I mentioned that we looked at eating behavior rhythms. So this is a video that shows our setup for the way we measure that. So we use infrared cameras to monitor the mice 24 hours a day. And we are then able to basically quantify each time there's an eating event. So you can see right now the mouse is eating, and we're able to use that to see, find the peak time that the mouse is eating and a few other measures of the rhythms. So here I have a few different measures of the circadian rhythms of eating behavior. So on the left here we have phase, which is an indicator of the timing of the peak eating behavior. So on the left you can see 12 to 24 hours. This is in Zeitgeber time, so 0 is lights on, so 12 to 24 is lights off. And since rodents are not, specifically mice, are nocturnal, they should be eating more in the dark period than in the light, or in the dark phase than in the light phase. And you can see here that something interesting we found is that the GPER1 knockout mice actually had a slightly delayed phase as compared to the wild type mice. So this wasn't something that was necessarily expected, but circadian one form of circadian disruption is shifted phase of rhythms, and that can contribute to a host of other problems. So we'll definitely look more at that. And then the amplitude on the right measures the robustness of the rhythm, so how strong it is. And we see that on low-fat diet, the GPR1 knockout mice actually had a higher amplitude rhythm, and this pretty much disappeared on high-fat diet. So one thing to note about the figures on this slide is they don't have any error bars, and that's because the sample size was really small. So a future direction is, of course, to increase the sample size. But even with just a few mice in each group, it's interesting to see these patterns. And then the other type of circadian rhythm that we looked at here was locomotor activity rhythm. So again, you can see on the left the phase measured in ZT. And you can see again that the GPR1 knockout mice had a slightly delayed phase as compared to the wild type. There is a little bit less of a difference than with the eating rhythms, but this group does have a little bit bigger sample size. So it's interesting to see that that difference is present with a larger sample. And then we also see in the amplitude on the right here, the amplitude of the locomotor activity rhythm is also slightly higher in the GPR1 knockouts on low-fat diet, and then that difference essentially disappears on high-fat diet. So our preliminary conclusions here are that GPR1 knockout female mice gain more weight than wild-type mice on high-fat diet, and that they also experience delayed phases of their eating behavior and locomotor activity rhythms. Of course, our future directions are to increase the sample size, and we also hope to look at the molecular tissue rhythms, which I mentioned earlier. And we look at a number of other metabolic measures like fasting blood glucose and body composition or adiposity. So before I conclude, I just want to say thank you to the entire Pendergast lab, especially to Dr. Pendergast and to Bucola, who have helped me so much. And without them, this project would definitely not have been possible for me. So thank you. And with that, I will take any questions.
Thank you. If you're just joining us, please enter any questions in the Q&A feature in Zoom. And while we wait for those to come in, it sounds like you have had a really wonderful experience in your lab. And I was wondering if you could speak briefly to kind of any highlights of your research experience here at UK.
It's hard to pick just one, but I really enjoyed my time in our lab, and something that I really like about it is that all of the undergraduates in our lab have our own projects essentially. So I know a lot of other labs, undergraduate students work with graduate students and like kind of help them with their work, but Dr. Pendergast has given us all the opportunity to have our own projects and work independently, which really has given me a good sense of what research would be like on a more advanced level. And it's also really exciting to, you know, go every week and weigh my own mice and check their food and things like that. So I really enjoyed my experience in this lab.
Fantastic. And a question that we just got in through the Q&A is, if eating phase is delayed in control mice on HFD, do they also gain more weight?
So that's a good question. We haven't looked specifically at, well, I should say in wild type control female mice, the phase is, if it's delayed, it's very marginal and doesn't cause any effects. In male mice fed high-fat diet, which do experience circadian disruption and diet-induced obesity, phases of several different tissues on the molecular level are delayed, and eating rhythm, it's really hard to determine a phase because it becomes pretty arrhythmic. So we don't have a very good model of a wild type mouse with a delayed eating phase to see if that causes weight gain. But we definitely know that shifting phases of other aspects of other rhythms can exacerbate weight gain in mice. And we know that basically the lack of major change is something that protects the wild type mice from weight gain and other metabolic issues.
Fantastic. I have an additional question. You had mentioned that folks in your lab had the opportunity to choose their own projects, and I was wondering what drew you to this particular project in question.
So research that was finishing up just around the time that I joined the lab by the graduate student who I've worked really closely with, and she's helped me and taught me a bunch of things, looked at basically what was protecting the circadian rhythms in female mice, and she showed that estrogen was the factor that was protecting them. So the next step of that was to look at the 3 estrogen receptors that have been well characterized so far. GHER1 is actually the least well characterized. It was most recently discovered. So that was kind of the next step of this overarching project. I've always been interested in physiology, and more recently I've become really interested in endocrinology. So looking at the effects of estrogen signaling and sex differences are interesting topics to me. So I was really happy to be able to work on this project specifically.
Fantastic. I'm just going to give it one more minute in case any final questions come through the Q&A. Okay, and with that, I want to thank you so much. This was fantastic. And I believe that this is our final presentation of this session. Let me just make sure that's the case.
Yes. Thank you all. Great job to all of our presenters. Thank you, Asia, for hosting and moderating session 2. And session 3 starts at 1:30 with Chalgren Fellows. So that's an interdisciplinary group. And again, thank you all. Great job presenting. For the faculty mentors that are in attendance, thank you all so much for your dedication and support of undergraduate research.